So, hi, hello everyone again. Yes, stream is up and running. Um, and welcome again to another Saturday Microscopy live stream. And yeah, today it's a little bit Christmassy, some beautiful, tiny little colorful stars here on the side. It's of course vitamin C on the polarized light. I'll be doing a little bit of that. And like every year, a little bit of a tradition. I want to put again yeah, a fir leaf. Here it is. Yeah, this year under the microscope. I've been trying to cut it into small sections. Not so easy, really. But I'll give it a try today. So these are basically the two main things that I would like to do, and I would like to, of course, as always, say hello all around the world, and of course. Yeah, happy holidays if you're celebrating Christmas as well. Just a little bit of housekeeping for me to do here. Yeah, I see that people started to join in the chat again. So hello from North Carolina, the USA, from Austria, from United States, UK, Netherlands, United States, Vietnam. Yeah, a couple of words in Esperanto, which I'll try to answer later. um, uh, Yes, there's a question here. Okay. Ah, yeah, there's somebody who wants to send me something. Yes, you can send them to me. Yeah. Hello from Germany. Okay. From Spain, California, from Poland. Okay, so yeah, basically, what I want to do today is I just want to show you a little bit of how I made those beautiful crystals here. I mean, I've made already plenty of videos on those, but still they keep on fascinating me over and over again. And there's some beautiful, more or less beautiful. Well, this region here is not so beautiful. Yeah, but this is in polarized light, and you do not need a special microscope or anything at all. It's actually one of the easiest ways to do a little bit of bring a little bit of color into the microscope, and you just need polarizing filters, which I want to show you. And yeah, again, here not so nice, but over here it looks kind of nice. Yeah, and that's vitamin C. I mean, I did already make crystals also with other substances, but vitamin C always works quite well. So I'll try to do that later, and I want to start out today with a little bit of a difficult one. Okay, I want to start off with a fir leaf, and I would like to try to cut that yeah, for microscopy. And it's a challenge, okay? And it's as I mentioned, a little bit of a how do you say, a Christmas tradition, yeah, where I am basically, where I try to basically always every year try to prepare that. Yeah, so I got over here, look, yeah, from a Christmas tree. And I would like to put that under the microscope, obviously, right? To cut it across, this is the difficult part. I also put some of them into alcohol a few hours ago to take away a little bit the green pigment, hoping that this might actually work a little bit better. Okay, don't know. Right, so we're gonna see. In any case, if you are new to this format, you can please feel free to ask any questions in the chat, but please do indicate at Microbehunter Microscopy or at Microbehunter or at Oliver, so that it's more easily possible for me to find your comments. Okay, so let me just occasionally I will also pause the stream. I'm not gonna go away, but I will occasionally pause it as well in order to simply read the comments as well. And yeah, there's 39 people who've joined in, huh? It's quite nice. Okay, so how are you gonna process this here? That's a little bit the question. Obviously, it's too big to be directly put under the microscope. Stereo microscopy is of course possible. I mean, that's usually the way that I start off with. Simply put it under the stereo microscope. But what we have to do is we have to cut it, and I'm gonna show you two ways on how to do that. Okay, yeah, one is with a microtome and one is without a microtome, and I'm going to just show it to you under the stereo microscope first. Yeah, here it is. Yeah, and you can see that. Yeah, more or less nice and fresh fir tree. Yeah, and big problem. They're way too thick and way too dark to put under the microscope, right? So I got to do something here, and that's something I would like to try to do. Okay. So a few more people have joined in. Hi from Poland, from the UK. Okay, greetings from Canada, and of course also holiday greetings from here from Austria, where I am right now. Well, I'm going to show you now one possibility of actually cutting that, and which actually does work. Not perfect, but it does work. Yeah, you can see that. Oh, a little. This is an insect that fell into the water here. Yeah, I have to just show you the one way that actually works. Kind of, I say, and that is you simply take one of those. Leaves here, we do not say needles. We just say these are leaves of a coniferous tree, and we just basically tape it on here. Yeah, and you just take some regular sticky tape. Okay, and you just try to stabilize it with some tape, right? Yeah, and that's pretty much it. Yeah, and then you take another slide. You go right on top here and you really press it down, and then you need some kind of a very sharp razor blade. And as always, if I hurt myself, then I'll put again some blood under the microscope. Okay, and yeah, you see this is also a little bit how do you say unstable, so I might also actually tape down the slide on here as well. Okay, I know this is a little bit improvised, but yeah. But if it works, it works. And what else do you need? You need also a, how do you call this? A Petri dish. And I'm going to add a little bit of water because this is the Petri dish where I'm going to be dropping in the slices, the sections, right? And you take a more or less reasonably fresh razor blade, hoping that you're not going to cut yourself. And what you do is you really have to press down on the top slide, and you basically just, you cut across. Okay, and this is the first one which you discard, and then you really have to move it over a very tiny amount. Yeah, a very tiny amount, and you cut it again, and you really have to press the flexible blade against at the top slide, right? Yeah, and then you press down as well. Yeah, okay, you press down as well, and you kind of hopefully are able to get a small cross section. And I actually see that this cross section is already still way too thick. This is actually a pretty bad one. Okay, so the basic the basic rule is a trial and error, and the more that you have, you see that the surface tension made it to float away. The more that cross sections that you have, the higher the chance actually of having one that's sufficiently thin. Okay. Yeah, so that's, I see that more people have joined in. Okay, greetings from Canada. Jess is here again. Hi, from Scotland, from the Netherlands. Yeah, as I'm saying, I'm just trying to try my best to make some of those, cross sections here. And as I just said before, is you gotta go quantity, because there is a certain statistical variation that you have here. Yeah, and you kind of hope that at least a few of them are going to be how do you say, thin enough to be useful for microscopy. I think I might need a tweezer, and they dry out quite quickly, obviously, because they're fairly thin, and that's why you put them into water, right? Yeah. Okay, there is a comment here already. I love this method with the cover slides. It works very nice for mushrooms too. I never tried it with mushrooms, like cross sections from the lamella apothecium. Okay, very, very interesting. Never tried it with mushrooms. What I'm going to do, however, now is the following: is I'm going to actually take this away, and I'm going to put it under my stereo microscope, and I'm going to show it to you also under my stereo microscope. But I do have the problem that I'm going to disappear now from this. You can still hear me, right? But you won't be able to see my face because I have to move over, and I'll take the microphone along. But it's a little bit. Let's see. I don't know if you're able to. Yeah, and I'm now going to switch over under the stereo microscope, and then you should also be able to see it there. Just a second. Yeah, and do you see this? Is here a little bit flexible, so I also have to tape that down. just to make it a little more stable. And yeah, now this is stable, and I also have to tape down. I have to, first of all, I have to find the, here it is. Okay, see, I did not cut it quite straight. Okay. So, and I'm going to use another tape to fix the slide in place. Okay. Here as well. That's now reasonably stable. And I'm now going to disappear from the image, so you're not able to see my face now, okay, because I'm moving over, but you're able to see what I'm doing here under the stereo microscope. And I don't like the arrow in the corner. So let's remove the arrow as well. Here we go. And let's have a look if this works a little bit better. Usually it does work a little bit better because I'm able to adjust the thickness a little bit better, but we're going to see. Okay. So. Okay. So I'm again making several cuts here. I just see that the camera is not properly adjusted. It's a little bit, as I'm looking through the microscope, it's a little bit out of focus. So I did not properly adjust focality. Yeah, but again, as I mentioned before, we have to go a little bit quantity, and we have to really press down. I have to press down the slide really to make sure that the pine leaf, the fir leaf rather, doesn't move. And then we kind of hope that at least a few of those here, that at least a few of those are thin enough. They're probably not, yeah. But I just want to illustrate this, yeah, to you. So I think I've got enough now, and we're just going to directly put them under the microscope. Okay, let me see. I have to loosen it up again here. See, everything has been taped on. I'm really scared of those razor blades because last time, or 2 weeks ago, if you remember, I really hurt myself, and then I could put my own blood under the microscope. Okay. And what I also found out, especially when making those thin cuts, you really gotta make, the tolerances are really small, so you really have to make sure that the slide and everything is properly immobilized, so really taped down and everything. Okay, so I'm going to now change again to the desk view. Here we are. Okay, back, here we are back, and you can already see that look significantly brighter than those over here. So what I'm going to try is I'm just going to try to directly make a slide. I need this here, and where is this? Here we go. I just take a little bit of water. It's just regular tap water. Put some on here, and I'll simply try to transfer a few of those or all of them on here. The thing is that if they are thinner than wide, then they will be flat, and we're able to see them from the side. If they're too thick, yeah, then yeah, we're not able to get them on the edge, right? Okay, here it is. Yeah. And now I'm just going to do it the very cheap and easy way. I'm just going to drop a cover glass on top, and we're just going to take it from there. So, again, this is a little bit just like in autumn. I usually make those the leaf veins very thick, and at Christmas time I usually always cut, yeah, the leaves of a fir tree, of a Christmas tree, Yeah, we're gonna take it from there and we're gonna see how well this actually works. Yeah. Okay. Yeah, there are a couple of comments here. Okay. Uh, yeah. I love the method with the cover slides. It works very nice for mushrooms too. Reminds me of chopping. Yeah. Yes, potassium ferricyanide crystals are very nice as well. If I may ask, are you a mycologist or similar? Oh, okay, that's not for me. Okay, that's not for me. Yeah. In German, Hobby mycologist. Very interesting. Pilzsachverständiger der Deutschen Gesellschaft für Mykologie. Oh, okay, so that means if I have a question about some mushrooms that I find and I'm not so sure whether I can eat them or not, then I know who to contact. Okay, so I'm putting out the old ones here, the old slide and this one over here. So let's have a look at the scope again. Okay, it's way too bright. I'm gonna put this in. Of course, it has to go into focus. Yeah. Yeah, okay. I mean, not ideal. This one is fairly thick. But you see that these ones here are fairly thin. They're a little bit torn apart. They're a little bit torn apart, but actually, yeah. Yeah, these are darker over here. You can see that the air bubbles are emerging. I could try to destain them a little bit by placing them into alcohol, right? But let's go up a little bit. Yeah, up. So, yeah, one of the nice things, let me, you see, with my age, I already have to put on my reading glasses. So there are usually always a couple of interesting things here. So first of all, usually what I do is when I get a cross section like this, I usually try to focus through the cross section to see how many cell layers I was able to get. So for me, that's sometimes a little bit of a challenge that I do with myself. How thin can I make those cross sections, right? And right now it does look that I am, have cross sections that are approximately 1 cell layer thick. Yeah. And now I'm going to show you how I do that. I'm going to turn on the arrow. Here it is, and just have a look at at this cell over here. You see over here, this dark on that's the cell wall, obviously, right? That's a very prominent structure also over here on the side. That's the cell wall. And let's just have a look at the cell wall, at the dark cell wall, as I go out of focus. You see? Yeah, it's there, and the cell wall becomes blurry, of course, but it basically stays there where it is. So there is no other cell wall going into focus, like a layer above or below. And this actually means that I'm 1 cell layer thick. And if it becomes blurry and another one goes in focus, then I have 2 cell layers. So that's actually, in that sense, maybe estimated thickness maybe around 10 micrometers, because that's the thickness of a cell, right? Also the fact that over here it's kind of torn open, that's a pity, but that's actually a sign that I'm cutting pretty thin, right? Okay. Yeah, so that's quite nice. And also let me go up a little bit more with the magnification. Yeah, of course, what you're able to see in here inside the cells, those green dots that you see, of course, chloroplasts and so on. Yeah. One of the nice things that I sometimes do, and I don't know if this is now also going to work if we were patient. I mean, I don't want to wait now half an hour or so, but you see those bubbles over here, right? That's an air bubble. And sometimes what you're able to see is that you're able to see that it grows over time as the cells continue to do photosynthesis, and then oxygen will start to build up. Yeah. So occasionally, especially when you've got a water sample with algae and so on, sometimes actually those bubbles will continue to grow. Yeah. And then you can put it on the time lapse. I don't know if this is not going to work here so very well, because the cells are kind of damaged as well, right? Also interesting to see that, of course, the outer layers here, are not green. Yeah. The upper epidermis, for example, is contains no chloroplasts, allows light to go through. I mean, over here, it's kind of interesting to observe this here. Do they have stomata and internal hollow spaces? So basically, if you just look at the thing here, the overall structure is to a certain extent similar, because on the top surface you also have this palisade layer, the palisade mesophyll with the vertical cells, and down here. However, I have to tell you, there are those over here, the so-called the spaces. This is called a resin canal. There is a one on either side. Okay. Yeah, these are called resin canals, and honestly, it would be yeah. If you look at on the yeah, they have to have some way a possibility for gas exchange. Yeah. So one way to actually find that out is the following. You know what? I will try this. Why not? Why not try this? I don't know if this is. I don't know if I have enough time to do that. But one way of actually testing the surface is the following. You take a leaf like this here, and you take some glue. Just a second. Yeah, this one unfortunately is already empty. Yeah, that's yeah, a glue that you can take. I'll just use a water-based glue. And what you can do is you can make actually a so-called a leaf impression by actually applying a little bit of glue on the bottom side and then letting it dry and then peeling it off. And then you get the impression and you're able to see the surface texture and the presence of stomates and so on. Oh, does this work? It's stuck. It is stuck. Okay, now it works. Okay. Let's do that. Yeah, but this will take a little bit of time to dry. Still stuck. Here it is. Okay. I don't know if this is yeah, if this is going to dry by the end of the yeah. But what I could do is I could put it on a warm plate. I'll put it over there. Yeah. So, and then we're able to see actually the bottom. Or another possibility of what you can do is the following. This also takes quite a bit of patience. Is you actually try to carefully remove. You know. Oh, let me take another one here. The majority of the leaf by cutting it off and then kind of putting just the lower epidermis under the microscope. This is difficult now to. Usually by cutting it diagonally, and then there's a little bit of a flap of cells here, and let's put this under the microscope. Okay. This is of course way too thick. I'm well aware of that. Always water to prevent it from drying out. And we give it again a little bit of trial and error and hope with a little bit of luck that we're able to see how the lower epidermis looks like. Hoping that I actually got enough here. So let's put the other one away. So again, the scope view, I have to find it. Here we go. And of course into focus. And then we have a view of the lower epidermis here. You see that? Yeah, it's pretty fairly thick here. And here I've basically cut open the cells. And yeah, but it's a little bit difficult to see. Yeah. But that is now basically the lower epidermis. And over here, yeah, the layer starts to become thicker again. you can see that we have now also, of course, here cells where essentially there's chlorophyll in there. Okay. Yeah. So this is kind of the, yeah. Let me quickly read a few things here. How is the structure of a fir leaf different? I mean, it is a conifer. I mean, there are certain similarities. For example, I mean, obviously the overall shape. But what you have is you have, of course, also the palisade layer. As we've seen, you've got a layer of more irregularly shaped cells. In the center of the leaf, you have the vascular canal, the vascular tissue. So overall, I mean, of course, the overall structure is similar. Yeah, because and they are obviously officially, and I wonder if those tiny little dots that you see, if these are actually all stomates or not. But I cannot go higher with the magnification because it's so thick. Yeah. So yes, transparent nail polish. Just a second, I have to see if I have some. Ah, yeah. Okay. Yes. Thank you for the recommendation, because this might actually work a little bit faster. So what I would like to do is the following. There's been the recommendation, why not use transparent nail polish? Because nail polish is a little bit also faster drying. So let's give this also a try. So what I'm doing now is just also taking the lower side here. And this is, and you just put a little bit of this nail polish on here. You allow it to dry, maybe a little bit more, so it's becomes easier to peel off. And then we're going to put the nail polish or the glue under the microscope. And because this would then gives you the surface texture, the impressions of the leaf on the nail polish or on the glue. Yeah? If you do, you can of course do this with all sorts of leaves, but the leaves have to be reasonably smooth, right? So if there are some leaves that are kind of hairy, then of course it's not so easy to peel off the glue later on. So let me put this away over here, and then we can have a look later. Okay, so that's kind of the thing. Let's see again what I would like to do now is the following. Where is the desk view again? Yeah, I would like to do now the same thing, also with those things here which I put into alcohol a few hours ago. They did start. I should have left them a little bit longer. They are brighter. But the alcohol will not only remove the water, but it will also remove the green pigment, the chlorophyll. And I kind of hope that this makes it a little bit more easily visible, the individual cells. Yeah. Do all green plants, with the exception of mosses, have a palisade layer? You see, it's always a dangerous to ask all, because if you find only one plant that doesn't, I just want to quote one of my professors. I mean, I don't know exactly because I'm not a botanist, so the word all is kind of a little bit dangerous to say, but I will tell you a quote of one of my biology professors long time ago, and I find this quote quite useful. They told us nature is so diverse that if you start looking hard enough, you're probably going to find pretty much everything in nature, right? Unless it violates some basic laws of biology, pretty much everything you can find in nature. Yeah. And I usually go with that. So if we go by that assumption, all green plants, with the exception of mosses, well, you've got also the ferns, you've got mosses, you've got. Then what about water plants? You see, so this means I would have to say no. Yeah, the leaf structure is most likely not going to be the same. Yeah, so let's try it. This one here is now with alcohol. Okay, and this one is of course now a challenge. Again here, and the problem with alcohol, of course, is it's highly volatile, so it evaporates very quickly. And because of the alcohol, I'm also not able to tape it down as easily, right? So these sections here are indeed a little bit thicker, maybe. Yeah, that's way too thick now, this one. Yeah. Some are thicker, some are thinner. But you see, it goes fairly quickly. But they do look fairly thick, so I'm not quite sure of, whether I'm able actually to turn them to the edge, to the side. Let's remove the bigger parts here. This one I think is from before. Now I have to add again some water. It's going to, of course, react a little bit with the alcohol, with the surface tension and so on. And if they are too thick, then they are going to not go to the side. That's going to be then a little bit of a pity. But they will turn automatically to the side when they're thin. So they usually, of course, they want to lie flat, right? But if they're too thick, basically they're not going to lie flat, but you're still going to get only a top view, which is then, of course, not so nice because we want to see it from the side. So I hope that at least a few of those, yeah, will be okay. Yeah. So that is the one that has been now in alcohol. Okay, so there is now some de-staining has happened. You see, that's what I meant. Yeah, too thick, and we only get the top view. That's a pity. Let me go down with the magnification here, with the hope. See, they're all. This one over here is on the side. So this one must have been sufficiently thin, but honestly, yeah, sufficiently thin, still too thick and still too dark. So the bottom line, I think, is that those hand cuts, at least here, without a microscope, not good. You see, this one over here is on the edge, right? It's on the side. Yeah, all others are kind of too thick. Yeah. So this was, yeah, a failed, under quotation marks, a failed little experiment. Okay, so I want to show you, however, another method that I tried, which did work as well, and that is the use of this microtome here. And yeah, I just want to show this to you as well. Oh, you can't see it. There's view. Here we go. So here we go. Yeah. So how am I going to put a one of those leaves? Yeah, into the, first of all, this one is how it looks like. This one is a clamp for a table, and this, by turning this here, and you see that it kind of always locks into place, right? It moves this piston up and down. Okay, so if I turn it counterclockwise like this, or clockwise like this, then it moves downwards. The question is now, how can I put a specimen in here? And the instructions that came with this suggested that I use a carrot and squeeze it in like that. And I said, it's kind of messy. So what I've done is I 3D printed those little specimen holders. And those two holes in the center are simply for taking it out. And over here, you see these little notches that you have here. This is where I can put a specimen. And I would like to do that by putting in, yeah, one of those things in here. So what I'm going to do is I'm going to put this in here. See, it goes all the way in. And then here on the side, I'm going to put, yeah, one of those leaves here. Okay, and that's it. Yeah. So what I'm going to do then is I'm going to take another homemade little device. Yeah? It's again one of those. It's a razor blade in here, and I'm simply going to cut this off like this. Yeah. You can see it over here, and I might have to make this a little bit bigger, yeah, so that you see it better. Right, here it is. I'm going to now, yeah, basically advance it. Let's say 3 notches, hoping that this was enough. Not really. 1, 2, 3, 4. Yeah, and I'm trying to always cut it off. Yeah. Nope. Yeah, you can see a tiny, a very tiny one over here. So I'm going to save it right away, so not to lose it. So, okay, and I'm going to put it directly in here. Yeah, here it is. It's a little bit out of focus, as you can see. But I'll do another one. Yep, very small. And another one. Here we go. And another one. So 3 of them now. And let's have a look at those. If they're a little bit thinner or not. You didn't see that. I put them on here. Okay, so let's make it bigger. You see that they're already significantly brighter, right? And this kind of shows that the cut must be quite thin. So I drop a cover glass on here again. I feel that again there is not enough water in here. So, and let's give it a try. So, and where is the scope view? Here we go. Yep. Yep, here we go. Do you see? This is slightly higher magnification. So you see that, yeah, definitely much brighter. Maybe there's also one over here. Yeah, also much brighter. And you see these were only 3 trials. Okay, this one over here is already a little bit damaged, but yeah, these over here, yeah, that's, yeah, I think significantly better. So I'm going to read again some of the comments asked about the palisade layer, because I was wondering whether this is maybe an old evolutionary trait. Would not surprise me, because the palisade layer with the highly compacted cells here on the surface, of course, they're very dense because they catch the sunlight, which is of course at the top. Yeah. So yeah, it's not surprising that there are actually so many packed cells here with chloroplasts to catch the sunlight. Yeah. So, yeah, macro shot of the cut leaflet ideas. A macro shot. Well, what I can do is I'm just going to show you how this actually looks like under the stereo microscope. Yeah, that's how it looks like here, right? So, and this is with light coming from the top. And this is with light coming from the bottom. And as you can see, the contrast is not quite as good, because stereo microscopes are, yeah, as I mentioned already, designed for opaque specimens, so that which do not allow light to go through. And of course, they are already kind of a little bit transparent. Yeah. So, yeah. So, yeah. I get it. Yeah. So basically, using a carrot that's is also being discussed right now. Is you drill a couple of tiny holes into a carrot, and then you try to make cuts like that as well. Yeah. I mean, in the macro photography, leave with the UT upon it photo idea. I don't understand what UT means now. Okay. So that is the. So that's a little bit the thing here, and I just realized one thing. Just a second. Let's try the following. Didn't try that yet. I think with the pine or fir leaves. So here we go. Just a second. I'm going to go like this. This is dark field now. This actually looks kind of pretty. This is not dark field. I have dark field only for my 4x objective. Oh, this looks kind of okay. Yeah. You see, if I go up with the magnification and I'm losing the dark field effect, because this is actually not just a dark field filter or patch stop. This is actually the for my phase contrast, and at 4x it also works with as dark field. Yeah. Yeah, there's a comment here. I heard that paleontologists use microscopes for thin slices of rock samples, though I'm not exactly sure how they make these sections. I can tell you that. I've not tried it, but they're polishing. You glue a piece of rock on a slide using some epoxy, and then you kind of polish it down until you get a very thin layer of rock. Yeah. So it's apparently a very painstaking, time-consuming process. But I know that there have been some time ago some people also in the forum and also in this live stream who were actually experimenting with polishing down hard rock samples. You have to glue them first somehow on a microscope slide. So that they really are stable, and then you have to use very fine sandpaper to polish down the rock. Yeah. Okay. Okay. Jesse bought some sponge spicule slides. Got broken in the mail. Oh, okay, I got it. That's a pity. Yeah. Don't even glue the rock to the slide. You encase the rock in a block of resin, like with the insects. Okay. So okay. Yeah, because maybe if you glue it on the slide, then maybe the slide might break as well. Okay. So you're kind of encasing the hard brittle sample in a resin block, and then you're polishing down, grinding down the resin block. Yeah, so this is kind of the thing here. So what we've learned, or what I've learned, is still the making those hand cuts without a microscope. Stereo microscope gives you fairly thick sections unless you're really careful. With a stereo microscope, it does work a little bit better because you can adjust the thickness, and the easiest is of course with a microtome, because then that's when we get those results here, right? So I'm just and then later on we're gonna just try a little bit to see if this actually works with the leaf impressions. I don't know. We're just gonna be a little patient here until the glue starts to dry. Yeah. Some rock slides. It would be nice, but I'm worried about the transport from New Zealand. Yeah, as we've just heard. Yeah. That's indeed a problem. I mean, I remember that even when I ordered some slides from eBay, even though they're nicely packaged, but of course, ultimately it depends a little bit much how. Yeah, sometimes they were in very thick padding. Yeah, because of course, especially historical microscope slides is of course if they break, that would be a real pity, right? Yeah. So I don't know. I think what time is it? 40. Time's passing. I would. Actually, I wanted to already several minutes ago. I would say that we're just gonna move on a little bit, and let's do a little bit of these crystals. Why not? Because crystals also kind of look nice. And as I mean, some of you already know that. So there's nothing new under the sky. But I just want to show you in any case, especially for those of you who are maybe new to polarization microscopy, just for the sake of completion, just want to give you a quick overview of what I'm doing. And I would highly recommend that, regardless if your microscope is advanced or simple, converting it to polarization is the easiest, or one of the easiest things to do. So that's option one. These are old filters, polarization filters from a camera, and I removed the ring here. These are so-called linear polarizing filters. They're almost impossible to get these days for cameras, because cameras use circular polarizing filters. I mean, they sometimes also work, but actually the linear ones are easier to use. And yeah, kind of difficult to get these days. I removed the ring on the outside which connects it to the camera. I simply took a saw and cut it open. Yeah, and these are made of glass, and the actual polarizing film is actually sandwiched in between the glass here. Has the advantage that easy to clean, solid. Yeah, nothing special here. Just to show you, if you have those filters, and if you rotate, it's kind of difficult to see now because it's kind of dark. So let's use a white piece of paper as a background. Yeah, if you rotate them, then you can see that in the crossed position, then it becomes dark. That's what we want. And if you put now some crystals in between here, you can see that the crystals actually appear again white or bright. And so that's basically the thing. Now getting these filters is you got to be a little bit lucky these days, because the modern filters, as I said, are circular polarizing. However, there's actually a cheaper way, and you get yourself some of those here, right? eBay? No, not eBay. Amazon, right? And pretty cheap, made of plastic, and you can cut them into any desired shape that you want, like I've done here. There is a protective film on the outside, which you have to peel off. Yeah, but I've been playing around with these things here. Okay, and it works pretty much the same way. You can put them into a crossed position. You see that they basically become dark, right? So this is a really easy way. Okay. Cheaper on AliExpress. Yes. Yeah, AliExpress. I've bought quite a few things already, microscope related things over AliExpress, including microscope objectives, eyepieces, camera adapters. So far it worked well. The only problem is sometimes you just buy stuff and it doesn't work. Like this weird DSLR camera adapter that I once ordered. But generally there shouldn't be a problem. So far all of the products arrived without problems. Yeah, there has not been any product that kind of disappeared on the way. Yeah, so in that sense, Avatar in IMAX 3D. Yes, it is possible indeed to use also those 3D glasses, the paper. However, be careful that sometimes they're also circular polarizing and not all of them might work. You have to find the correct orientation, right? So, You mean Avatar in IMAX 3D? Are you talking about the third Avatar? The one that lasts over 3 hours. Wow. So what I would like to do now is just show you what I'm doing. I place one of them directly over my over my microscope lamp, and the other one over the slide. And what I would like to do now is instead of doing this under the compound microscope, let's try it over here. Then on here goes the vitamin C. Yeah, you see over here. Let's go a little bit out. So that's the one that you just have seen at the beginning of the and then the other one goes across. I have to turn it right now until to make sure it's dark. Yeah, you see it's a little bit dirty here. And then let's zoom in a little bit. And I have to refocus. Yeah. Nope, not from the top. I need it not from the top. I need it from the bottom, of course. Why does this not work from the bottom? Here we go. Yes, okay, that's now with light from the bottom. Yeah, and you can see that there are now the colors. Before I had the wrong lamp. Yeah, so that's basically how it looks like under the stereo microscope, right? And this is exactly the one that we had just before, under the compound microscope. Yeah, so these vitamin C crystals look quite nice. And what I would like to do now is I just want to show you how I prepared them, right? So basically we're gonna go to the very beginning, and I'm just gonna show you how this works. Okay. And again, if you don't know what to do over the Christmas holidays, if you have holidays, then this is something I recommend because it's kind of fun. It's almost foolproof. It's fairly easy to do that, and it's kind of fun. And what I need is I need some slides. Oh my gosh, did I already use all of my slides? Almost. Here we go. And what you do is the following: is I'm going to put all of the leaves away. You need, of course, something like a tiny little beaker, or any. You can of course also dissolve the vitamin C directly on the slide. That's possible, but I like to, do it differently. Okay, so you take a little bit of water. Number one, you take a little bit of vitamin C. We are not talking about vitamin C tablets, which contains a lot of filler material. We talk about pure vitamin C, pure ascorbic acid. I bought this some time ago in a drugstore. Just walked in and said, I want to buy some vitamin C, and this is what they gave me. Yeah? And you take, it's just a pure white powder. Yeah, and I tried this before, and for this amount of water, around 2 of these scoops are enough, and this will give you a lot of slides. Okay, and you just basically dissolve it. You don't have to dissolve it completely. If there's a couple of crystals left over that are not dissolved, actually it can look kind of pretty as well. Okay. Are you using bright field or polarization in your microscope? My microscope, okay, as it dissolves, I'm going to try to answer this question, okay? So let's have a look at my microscope. So this one over here is dark field. This one over here is regular bright field, okay? No polarization, okay? And what I'm going to do then later is for the crystals, I'm going to use polarized light. Yeah? So this is basically, and what I also have is I have something called a DIC. And that's now, DIC. And DIC is a pretty fairly advanced technique, which also uses polarized light. And you can see that the background looks red, and it can change the colors around as well. Yeah. So if you ever see those videos that I make with this bluish background or so, then you know that I'm using DIC. Yeah. So this is kind of there are also other ways how I can adjust it. Yeah. So that's basically what I have. But normally, just for regular observation, you just use bright field, then you get the natural colors. And what I'll be using now is I will not be using DIC obviously, but I'll be using the polarizing filters. Yeah. No. The question is, does that mean I won't be able to see those colors if I don't have polarization in my microscope, even with polarizing filters? No. You convert your microscope, your regular standard bright field microscope, you convert to polarization. You don't need any fancy stuff. So it works with the simplest kind of microscope even. Okay, so that's what I'm saying. You don't need anything fancy. All you need is you need 2 filters, or one of them which you cut in half. Yeah, works with any bright field microscope. Yeah, and that's what I'll be also doing here. Okay, I'll be using that's why I also kind of showed it to you using my stereo microscope just now, which basically has nothing. You just sandwich the specimen between those 2 filters. That's all you need. Yeah, so it's one of the easiest. How do you say, the easiest conversions? Yeah. So I just want to reassure you. Yeah. So the vitamin C has now dissolved. Because I'm impatient, I'm using a coffee warmer here, which you don't have to do. I'm always using this, especially for those live streams, because otherwise there's simply too much waiting time. Yeah. And what you do now is you simply put a couple of drops here, on the slide. Sometimes I do add a bit of alcohol simply to break the surface tension a little bit, because sometimes you see it likes to stay in a drop, which is sometimes a bit annoying, because then it might become a little bit too too dense. So sometimes what you might want to do is the following: is add a little bit of a drop or so of alcohol, which is going to evaporate anyway. This was not a good idea. Let's do it like this. So that's alcohol now, okay? And see, if I add now a little bit of alcohol to the whole thing here, you see that it immediately starts, yeah, the surface tension starts to break. Yeah, and the drop starts to spread apart a little bit. Not always necessary, okay? But I'm just mentioning this, so, yeah, so that you can experiment a little bit more. Yeah. So let's have a look. Yeah, okay. The question is you've changed a format of your channel. I really like all of your live streams. What happened with the previous streams from the other channel? I don't see any of the live streams. Ooh, well observed. So as I put this on here, this is kind of very hot. Okay, as I put it on here, you have noticed something indeed, well observed. I need to tell you a little bit, and I might put them back on. It's correct that the other channel, I removed the live streams. So the recordings of the live stream. So I need to clarify this as we wait. I've got two channels. I've got this channel over here, which are called Microbehunter Microscopy, and I've got another channel called simply Microbehunter. And the other channel I used to, up to a couple of months ago or weeks ago, I used to do a lot of mostly of the microscopy stuff on the other channel. And basically this channel that you're watching right now, I did a lot of optical stuff, technical stuff, hardware reviews, and so on. And what I've seen over the last couple of months is that the number of views really dropped down. So I don't know what's going on. People, didn't watch so many videos anymore. I was just sometimes only in the hundreds. There used to be thousands of views per video before, a couple of years ago, and then it dropped all the way down. So as I said, YouTube somehow changes in unknown ways. And so what I've done therefore is the following. I moved all of the microscopy related hands-on stuff like we're doing right now to this channel here. And I'm trying to convert the other channel, to pivot the other channel into a how do you call this, microbiology explainer channel. So I try to separate the topics a little bit more. So if you watch some of the recent videos on my other channel, the Microbehunter channel, you're going to see that I've done some videos on food safety, a little bit of theoretical microbiology and so on. And I'm trying to still find a little bit the correct or the best format. But all of the hands-on demonstrations and live streams and hobby microscopy stuff is now on this channel over here. And for this reason, I did not delete them, but I temporarily put the live streams and also all of the shorts that I made for the other channel. I put them I unlisted them because I got the wrong subscribers for the other channel. So this is a little bit something simply to kind of clean up the channels a little bit. But I might, if you're interested, I can turn them back on again. But one of the reasons why I lost so many views and subscribers is because I actually had the wrong subscribers. Yeah, a little bit. Yeah. So can you show soya sauce in polarization? Oh my gosh. Yeah, I could. But I don't have any soy sauce here with me. So maybe somebody can bring me some soy sauce. Maybe the door is going to open in a minute or so to get me some soy sauce. I mean, I did some weeks ago, I did actually put some soy sauce under the microscope. They look very nice, okay, as well. There are lots of salts and amino acids and so on in there. Yeah. So let's try this again, okay? And I'm now going to do the following. I'm going to put now a polarizing filter. I'm going to turn off the DIC. I'm going to now put a polarizing filter in here. I put the vitamin C on top. Okay. It's still kind of very bright. Okay. So, and these are already the first crystals forming. No, not yet. Yeah, not yet quite. Ooh. I have to check where's this interesting stuff. Oh. I have to see if this actually, and I have to put now another polarizing. Let's turn down the Yeah, here that's already a crystal forming, and I'm going to put now one of the filters on top. Directly, I'm gonna drop it directly on top here. I have to focus it. Okay, and I have to make sure that the bottom one is in the crossed position until the background is all dark. Yeah, and that's already a first crystal forming, but I don't see it grow very much, which is kind of a pity. So maybe I didn't quite get the concentration right. Yeah. So, so what I'm gonna, maybe it's growing here a little bit. Usually if this happens, what I do is I simply, maybe I did not get enough, maybe the concentration is not high enough. So what I shall do therefore is I'm going to try to add a little bit more of this again on a, you see, it stopped crystallizing. Yeah. So, okay, I'm gonna show you where I put the bottom one. Let's do the following. I'm going to now flip over the camera. Just a second. I'm going to show you now where I put the filters. Everything's going to be a little bit upside down now. Let me see if I can actually flip it around here. Here we go. Okay. So that is now the lamp of my microscope. I can't get it direct. I mean, okay, you get the idea, right? So that's the lamp of my microscope. Yeah. Now, if this is Köhler, forget about it. I open it all the way. And one filter goes on the bottom. Okay. So look, you can see what happens. This filter I just put on here. Yeah, it's the larger one. I use the smaller one here. I just drop it in here and I can turn it. Okay? That's important, yeah? That's it. Yeah? So what however, that's what you're probably gonna do. And if you don't have a filter like this, what you're gonna do is you're gonna take this here, okay? A filter, you remove the cover, you remove the protective cover, and you put it down on here. Okay? That's exactly the same. You take one of those here, you remove the protective cover, you cut it into a nice size, and you put it on here. Yeah, that's it. Thank you. Can you bring me some soy sauce? Okay. So I just got the questions. And the other one, the other filter goes on top. Right on here, and of course there is not a problem that the filter is going to touch the crystals, right? So maybe you want to make some kind of a spacer or so, but I'm sometimes just a little bit lazy and I just drop it on here like this. Yeah. Thank you very much. Here's some soy sauce as requested. So let's I'll do some soy sauce polarization, right? And then what you do is you have to turn it in such a way that essentially the background becomes all dark. Yeah. So let's have a look at this. So where is the scope view again? Just a second. Yeah. So I have to turn now one of them. I'm just gonna. No, I forgot the bottom one. Yeah. So here we go. Yeah. And you see, I'm not turning the bottom one. Yeah. Until the background becomes all dark. You can also turn the top one. It doesn't matter. Okay. The bottom one might be easier to turn. Yeah, here the background is too bright. You turn it until it's all dark. Right? And again, if you don't have a glass filter, you use the plastic ones, okay? Yeah, and you just basically cut it nicely into convenient shape. Yeah, maybe you want to glue it on some, I don't know, some cardboard ring, a cardboard ring to make it more stable, whatever you want to do. But you can already see here that it actually did slowly start to grow. And let's go up higher with the magnification. Let's see. I think it is slowly crystallizing. It is slowly growing. And you know how I'm going to find this out? I'm going to turn on the arrow. I'm just going to put the arrow over here, right at the tip, and we're going to see, just to remember a little bit of where it is, right? Yeah. And if you flip one, it will turn into a CPL instead of a, only if you've got the circular polarizing filter contain a linear polarizing filter and a lambda fourth plate. So actually it's a circular polarizing filter is actually made of two of them. But these here are the simple ones. They work the same way regardless if you flip them or not. Okay, so they're a little bit easier to use. Yeah. So I'm just saying, yeah. Yeah. So if your microscope, for example, has a filter holder, a swing out filter holder, for example, then of course you want to cut out a circle and put it directly into the filter holder. I mean, obviously because it's much more convenient. And you can actually leave it in there if you want to. And if you want to have polarization, then essentially just drop in a filter on the top. Yeah. Yeah. So. Interesting, I found the film ones I get only work if they're placed the right way. Yes, the ones that, I mean, there are those film polarizing filters which are used for those 3D glasses. And those basically somehow are circular polarizing and where the orientation indeed is important, how you flip it. Yeah, so there are indeed apparently different types of these film filters, and I just got the regular linear polarizing ones because then luckily the orientation is not relevant. Okay, so you see, yeah, look, it's slowly growing. I just want to show you something else that's interesting, and we've already seen this happening over here. Let me show you. Over here, it all started to polarize. Not polarize, crystallize. I have to turn it again. Yeah. And then there's now some fun stuff that you can try to do, okay? Just want to show it to you. Yeah. Sometimes you see more colors, sometimes you see less colors. But what I recommend that you try to do is the following. I'm going to turn on now the desk again because this is, I have to flip it around again because from before. Look, I'm going to put it on here. Yeah, you see all of those white areas is where the vitamin C started to crystallize out. However, you're going to see that over here it did not crystallize yet. So I'm going to show you something interesting. What you can do is you can carefully try to touch it here. And sometimes this will start a crystallization process here. Do you see how it turns white? Yeah? You touch it and then it starts to crystallize. So the dirt and the cells of your skin, they act as so-called crystallization seeds. This is where the crystallization process starts. So you can actually, it's kind of sticky, but when you touch it, right, then after a couple of seconds, it should actually start to form some. That's why it slowly starts to turn white. Yeah, you should actually see some crystals forming there. Okay, so let's have a look at this here. Yeah, this is where I deliberately started the crystallization process. So again, let's put the thing on here. And then the scope view here. Ah, look. Yeah. Look, this is basically where I kind of scratched across. Yeah, and this is where now crystallization started. And you can, here, that's not nice because they start to grow a little bit faster. Can you see this is now in real time? Can you see actually the crystals expanding? Yeah, so let's turn off the arrows. I personally find this very pretty. I mean, I started doing this stuff like this long time ago with my first microscope, and I've made some beautiful pictures. Yeah, yeah. So just saying that this is vitamin C, very nice. And as we look on here, I'm going to now do the next thing because this was the request. And again, I know I've been repeating this a little bit, but it's still kind of fun. I just want to show you some soy sauce. Soy sauce. No, I just realized I don't have any more beakers left over. Just a second. I think I should buy myself again a couple of beakers. Okay, yeah, you get the idea. Look very quickly. Desk view. Where's the soy sauce? Same story. Soy sauce actually, honestly, if you can't get vitamin C, get yourself some soy sauce. Because it's Also very nice. It's easier. You don't need to buy it. You don't need to dissolve it. So here's my soy sauce. And generally the soy sauce spreads a little bit more easily than the, yeah, you can see somehow maybe all of the other things that are in there, yeah, break the surface tension a little bit. I think enough. And yeah, then you have to wait again for a long time, or you don't wait for a long time because you put it into your coffee warmer. Wait, let me put the soy sauce tip away. Okay, yeah, I mean, you see it on the top. Here it is. Yeah, here. Yeah, some bubbles there as well. Maybe they're gonna pop. I don't know. Yeah, they're popping. And you just wait until the soy sauce evaporates, and then you see again pretty nice crystals. I think that very different, but I think the soy sauce crystals, whatever substance is in there, some, I don't know, maybe some salts and some amino acids, maybe this taste enhancer, yeah, glutamic acid. They make really pretty crystals here. Would you show us your pictures the next session? Oh yeah, I've got lots of them. Oh, I've got like an explanatory way. I mean, there's not a lot to explain to the pictures. I've got a lot of them that I made. Vitamin C and citric acid and so on. I put actually some of them even up in my, hung them up on as pretty pictures. Yeah. Does vinegar have salts in it? Well, no. Vinegar is the acid, and you only make a salt if you add a base to it. And vinegar itself should not crystallize. Should it? It's an acid. It's no, of course. I mean, it's going to evaporate. It's I think what you need to do is you need to neutralize it first to make sure that the acid group it becomes charged so that it might crystallize. Yeah. So let me quickly read a little bit. Yeah. Let's have a look. Okay, so far no comments from me. Yeah, but you know what? It's 40 degrees. I'm gonna go up with the temperature. Let's go up to speed it up a little bit. I mean, you see on the side, it started to crystallize already a little bit here. So let's see. Let's go. So soy sauce generally is quite nice. I hope that this works also. So what other comments are there? Vitamin C is ascorbic acid. That's correct. Why not acetic acid? Because acetic acid is a completely different substance. It's acetic acid is is not even optically active. Ha. Acetic acid is not even, because it does not have a chiral C atom. Now I'm going chemical. Okay, so yeah, in that sense, yeah. What is this? No, it's okay. Okay, yeah, okay. Yeah, here, vinegar. Let's try it. Let's give it a try. I got some vinegar now, and let's put some vinegar under the microscope. Then let me see. Ooh, I'm running out of time, but it doesn't matter. I'm not gonna stress myself. So here's some vinegar. Never tried to crystallize vinegar. Cannot imagine that this works, but who knows? Who knows? It's getting too hot. Oh my gosh. Hope I didn't overdo that. Oh my gosh. Okay. Let me cool this down first. Okay. And let's put the soy sauce first under the microscope. So let's remove it. Let's remove the vitamin C. here is now the soy sauce. Maybe I've overdone this a little bit. Let's see if this scope. So that's the soy sauce. Filter on top. Oh, it's still. This was still liquid. Oh. Okay. Huh. Let me see. For whatever funny reason, the effect is not quite as good as I hoped it to be. Maybe there's still... Yeah, but you see the crystals over here? Let me... maybe I boiled it too much. Let me try the different one here. Here, it's a different polarization filter. You see? Yeah, see, it's still liquid. That's why. Dang. You see the crystals here? Okay. It didn't completely dry yet. So for this reason, I need to probably put it back again a little bit on the warm plate. Okay. And I'm going to try now the vinegar. So, and for that, I need a new slide. And here we go. That's the reason it was not... Yeah. Over here we have now the desk view. So here is the vinegar. Here it is. White vinegar. And let's see if it crystallizes. So, and over here I've got the... Now hopefully... It's hot. Need to cool it. So, this one over here. And the scope view here is the... Turn it down a bit. And on top I put now the... I need to clean this here. Because there is soy sauce now all over the filter. Yeah. Let's see if I can get this done here. Yeah. You can actually, it's kind of interesting. A second. Let's try it like this. Let's put this one away. Let's try it like this. Here we go. Yeah, this one actually looks a little bit already, I would say, disturbed. But normally what you can actually do is you can actually see those crystals grow quite nicely in soy sauce. Yeah, and actually they are quite pretty. Pure acetic acid solidifies into clear needle-like crystals at a temperature just below typical room temperature. Ah, okay, interesting. So you need almost pure acetic acid. Yeah. Okay, but you see over here, these are the soy crystals. Now, the only thing that kind of is interesting is, let's put this away here. Let's try this. Let's turn up the... Yep. Where is this? Yep, here we go. Okay. Yeah, so you get the idea a little bit. Yeah, you gotta play around. What about this one over here? Still did not dry. And what I would like to do as well is the following. Let's put this here away. Let's put this here away. And another thing that you might want to try, and that is also a possibility that I just wanted to show you, if you have a a glass polarization filter, instead of actually crystallizing it on a slide, you might also want to try to crystallize it if you have a glass polarization filter directly on here. Yeah, might make it a little bit easier. What you do in this case is the following: you can then take a little bit of the vitamin C solution and put it directly on the filter and crystallize it on here. See, this one over here is, yeah, and then, yeah, but then you have to again be patient a little bit. Yeah, I think I'm not gonna do that. I'm a little bit worried that if I put it now on the hot plate, that maybe the plastic filter, which is sandwiched between the how do you say, the glass, might actually become damaged, or maybe the glue kind of dissolves. So actually what I have to do is I have to kind of dry it at room temperature, right? And then when you do that, then you can put the second filter on top. Yeah, like this. Yeah, that would also be a possibility, but again, I'm a little bit worried about putting this filter here directly on a hot plate. Yeah? So I think we just stick to the, how do you say, the the glass slides. Where did I put the previous one? Here it is. Yeah. So this is again the vitamin C, and I just want to show you something else. What we can try to do. I'm going to remove this because this is sometimes also quite fun to observe. And what you now need is, again, the scope view. We reduce the light here a little bit. We now need to put, with a little bit of a distance, I'm going to put a spacer. And I'm going to put this now over the slide with a spacer. Okay, so that's the vitamin C again. And in order to do that, I'm going to use 2 more slides, which I put perpendicular across. I need to open the slide box because I would like to dissolve these crystals again. So I'm going to do it one over here, one over here. This one goes on top across here so that there is a little bit of a space. Okay. And what I can try to do, if I'm just able to reach this, let me get this like this, more light. So like this. So it looks kind of pretty. Okay, that was the one from before. I'm now going to try to dissolve it again by adding over here a little bit more of the not citric acid, the ascorbic acid, the vitamin C. I'm kind of hoping. Yeah, do you see this? How it starts to dissolve? Looks also sometimes pretty cool. Yeah, that's yeah, and then usually it stops after some time, and then it starts to recrystallize again. Yeah, okay, so but that's kind of the reverse process. Then the vitamin C solution is pretty saturated. Vitamin C solution is pretty saturated, and then it would start to recrystallize. Let's wait a little bit. Yeah, and because time is slowly running up anyway, so there are a couple of questions here off topic. What would you like for Christmas? Do you have all the equipment, or do you ever want, or is there something else? Honestly, with the equipment that I have, I'm pretty happy actually. Because what limits me is not is not really the equipment or the hardware. I'd like to have a little bit more time to do microscopy, honestly, right? I mean, I'm working full-time as a teacher as well. So the microscopy live streams that I do on a Saturday, of course, every week, that's kind of a regular thing that I do, also deliberately to kind of get my mind off work a little bit. But I would say a little bit more time. And I mean, I was actually thinking about buying, the company does offer how do you call this, an electrical way to control the movement of the slide, because if I now move it around, it's kind of a jerky, right, and it's not as smooth. But it's possible also to move it in a by using a motor, and then you can make very nice videos this way. But then again, I tell myself, it's getting all so advanced, and I think microscopy can and should also be a little bit simple. You just put stuff under the microscope and you enjoy nature, and you should not always, I try to tell myself, just go by the hardware, right? Having said that, I think that would be kind of nice, and I've been actually trying to make a few of those stepper motor things myself, where I can actually remote control the stage using some buttons, right? But that's a little bit, I would say, that's rather more on the fancy side, yeah. So I'm just reading a little bit. Yeah. Technically, you can even melt wood. Yes, and I remember that it has been a video on this somehow. Somebody has actually molten some wood, yeah, with a lot of pressure and friction. It is possible. Yeah. Yeah. You see, it's still dissolving. Yeah. So the capillary action kind of pulls all of pulls everything along. And there will never be two crystal patterns that are exactly the same. Yeah. Yeah. So that's heat wood without any oxygen present, and it will melt. Interesting. Yeah. So I do have, I don't know, I already answered most of the questions anyway, but there are a couple here. The position of the filters I showed. Off topic, any plans for microscopic mysteries to return? Would love for the answer not to be revealed until the next episode. Oh, that's actually an interesting one. Having maybe one episode per week. Okay, I need to explain this a little bit. Over the past couple of years, I've experimented a lot with different channels. Just for your information, there was also, or still is, a channel called Microscopic Mysteries, where I made a few videos using my stereo microscope, and you're supposed to guess what it is. I haven't been making a video for some years now, but this is kind of one of the things that you might want to check out, right? But I could, kind of include that idea a little bit. Maybe it's actually quite a nice idea. Maybe in this live stream, where at the end of each episode, where I show you an object under the stereo microscope, and you can guess what it is, and the next week I will reveal it. That would be actually quite a nice addition to the format. Yeah. Yeah. So I'm just reading here most of the comments I already mentioned. Okay. It's about the leaf anatomy. Yeah, I think I did already address all of them. Yeah. I have just looked at Amazon, and there are some microtomes which do not cost very much. I have no idea if they are any good though. Yes and no. Haha, that's a good point. That's a difficult one. I did make a couple of microtome videos as well. A lot depends really on how well you fix your specimen in the microtome. I can just show you something, just a second, just because you're asking. Let me show you. Here is one. Here is another. I'm just going to quickly show you two microtomes that I have. This one over here that I used before, I got that from Brunel Microscopes in the UK. So I did not buy this over Amazon, but in the UK, there's a company called Brunel Microscopes, and I've already ordered a few things from them. And this is actually one of their microtomes there. I'm quite happy with it, right? The only thing that I had to do is I had to make myself this specimen holder, because they recommend that you use a carrot, which also works. Yeah, but actually this is a very nice one. Yeah, and one of the cheapest ones that I got. I did not buy, I don't know where I bought this. Yeah, looks like this. There's a glass plate that falls off. But there is a clamp in here with a screw, and it kind of works as well. Yeah, better than nothing, right? But it does not look like the highest quality, but it does its job. Yeah, so, and then there was another one, which actually is of a much higher quality, but which I'm not using. That's this one here, much higher quality. But the problem is there is no clamp in here. And because there is no clamp, yeah, I have a problem actually holding the specimen. So what I have to do here is actually just like the one from the Brunel microscopes, I have to make myself a specimen holder or work with a carrot. Yeah, but this one actually is way more, way smoother and more precise. Yeah, it also comes with a nice clamp here, yeah, to mount it against the table. Yeah, so I'm just saying, I would say there is no, how do you say, no perfect solution. Currently, I am using the one from Brunel microscopes the most often, also because, yeah, it's nice and compact. Yeah, and another one. Let's look again over here at the. We're almost completely dissolved now, and this is a homemade one. Yeah, this one over here. This is a wash over here, and it's a little too bright. Yeah, with a screw on the bottom. Also works. So for plant stems, I've been using this one here. Yeah. I'm just going to, yeah. So basically, this is the dissolving of the vitamin C, and you know what? As a last thing, maybe I'm going to take it off, and I'm going to. You see, the vinegar did not completely evaporate for whatever reason. It's interesting. Yeah. So desk. Here is the vinegar. It's still liquid. Yeah. Let's test it a little bit if it is. Yeah, it's syrupy, almost like thick honey. Like thick honey. It doesn't crystallize. Yeah, I'll wait a little bit. But you know what? Just as a last thing today, over here, let's put the thing here back into view. How do I do this here? It's difficult. Yeah, let's put it back on here again. Yeah, you can slowly see that the water again is evaporating, and it's starting to recrystallize. So I'm reading again some comments. Oh, it's already 1.5 hours. Time to stop. But yeah, in a minute or so, just as a last little thing, let's put this again under the microscope. It's hot. And then I'll call it a day. And then I have to clean up my desk. So scope view. So here we go again. Yeah. Yeah, so you already see the next crystals forming, and here it's growing again, as you can see. Yeah, here we've got some recrystallization, and now that's again. Okay. So let me quickly read. Some off-topic conversation going on here. But I think, yeah, what I will do is I will have to refer you to continue your conversation either in the comments section or maybe over email or so. Okay. Yeah, check my emails. Yes, I got your, thank you. I got your video on to ever smaller about the spermatozoa. Thank you. So and I got also an email some weeks ago on some slime molds, but unfortunately I could not open it. So, but maybe the next time I'm able to show them to you. So yeah, so that's basically it, folks. I wish you a very Merry Christmas, obviously, all around the world. I think I might, of course, why not? I'm gonna next week is the 27th. Should work out, I hope, again for me. Yeah, I hope that yeah, I'll be able to see you again. And I'm going to call it a day for today. I'm going to call it quits and hope to yeah, see you again next time. Merry Christmas and all the best. Bye bye.